Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This is the second one in the genetics engineering unit and looks today at cloning and genetic modification. Now, this is what people generally think of when they're thinking of genetic engineering. The first video in this sequence was on artificial selection, which is a very, very ancient form of controlling the genetics of organisms to suit our purposes. But this really is pure genetic engineering, where we're actually fiddling with the DNA itself. This is from the GCSE curriculum, and as always, please do subscribe to my YouTube channel if you haven't done so already. Thank you. So if we're going to look at cloning, which is obviously an artificial process, what we need to look at first is the thing that we're comparing it with, which is the natural process of making a baby. Now, I'm going to stick with the example of humans because it's most relatable. It's very exciting thinking about the cloning of humans, but it's internationally illegal to clone humans. So this example isn't actually one that could be done, but the theory is the same. So we're going to use humans, but then we can switch around and think about any animal that we like. But let's think about the natural process of making a baby. The first key point is that it's done in vivo. Now, that's a good term to know. In vivo means inside the body as a natural process. It's going on inside. The opposite to that is in vitro, which is done outside of the body. And we'll talk about that in a minute. So making a baby naturally, it's done inside the body. You need a father and you need a mother. From the father, there's a sperm cell. From the mother, there's an ovum or egg cell. Those two fuse together at fertilization, or rather the nucleus from the sperm fuses with the nucleus within the ovum. So that ovum becomes a fertilized egg, which is a zygote. So that zygote is the very first cell that has the full 100% set of DNA. Now that zygote is going to multiply by mitosis. And mitosis is going to make a load of cells that start to organize themselves, start to differentiate, and we get an embryo like this one. Now the embryo in vivo, inside the mother, is going to grow. She's going to become pregnant. She's going to give birth to a little baby, to this little guy. I've marked it in purple because that's then the cross between the red and blue parents up there. So you can see that it's a cross between those two. It's 50% mother, 50% father. That's the natural process. Now in cloning, it's something different because a clone doesn't have a mother and a father to mix the two sets of DNA. A clone is an exact copy of an organism. So as a first key point, and I mentioned this term earlier, if we're now talking about somatic cell cloning, I've changed the title up there, the first key thing is that it's done in vitro. Okay, so that's why I've put a petri dish up there to show this is something that's done outside of the body. Also, it's not with a mother and father, so let's get rid of those labels. We could still have the man and the woman there because we might still use both of them. But if we are planning on cloning the man, let's say, the guy in blue up there, we'll just refer to him as the subject that is to be cloned. We want a copy of that person, okay? Now, we still need a female at the start of this process because the only cell that can grow into a new mammal, including humans, is an egg cell. So we do need an egg. So the woman over there, I'm putting it as the egg donor or ovum donor. So she's going to give her egg cell still. However, the subject won't give us sperm because the sperm will only contain 50% of his DNA. And we want 100% if we're going to clone him. So instead of the sperm, what we need from him is a somatic cell. Now, a somatic cell is just a normal body cell. All of your body cells are somatic cells apart from your germline cells, which are your sperm and egg cells. Okay, so any normal cell, could be a skin cell, doesn't matter. They should all contain 100% of, in this case, that blue guy's DNA. Now, step one that we need to do from here, once we've got those two cells, we need to remove the nucleus from the ovum because that nucleus contains 50% of the woman's DNA. Well, the woman's going to be nothing to do with our clone. We don't want her genetics. We don't want her proteins going into our clone. So the only instructions she's got is that 50% in that nucleus. So we get rid of that. Now we need to reprogram that egg. Well, we've got a full set of instructions to reprogram it with. So step two of this, we take the nucleus from the somatic cell from the man and put that nucleus into the egg. That egg now has 100% DNA instructions, just like a zygote does. But of course, it wasn't formed by fertilization and it's not a natural thing. So this is an artificial zygote. It contains 100% DNA, but the 100% wasn't 50% from the mother, 50% from the father. It was just 100% from the subject wanting to be cloned, put into an emptied egg to reprogram that egg, so that can now grow by mitosis into an embryo as a normal process. Now, this is where we do need to fall back on the in vivo bit, because you can't just leave an embryo sitting in a petri dish and it will grow into a human. The only place at present where that embryo can grow into a human is inside the womb, inside the uterus. Stage that embryo would need to be implanted into a woman so it could grow, she's pregnant with it, and she would be able to give birth to it. Now, that woman is called a surrogate mother. She's carrying the embryo, she's going to give birth to the child, but it's not genetically hers. She's a surrogate mother. She'll carry it and give birth to the clone. So look, the little baby, now I've changed to blue because that baby is an exact genetic copy of the man we cloned it from. And when it grows up, it should become a genetically identical copy of the original subject, of the original man. This man next to me here essentially is that man up there. Now, of course, if you've already watched the lecture of mine on the difference between genetic and environmental influence on variation, you'll know that this person is never going to be exactly the same as that person because their environment's going to be different. This guy might eat really unhealthily and become really fat, in which case he might be a really fat but genetically identical version of that one. He might go to the gym and lift loads of weights and become a really muscular version. He might grow up in a different country, have a different accent, a different language. There's loads of ways in which he will differ, but only in environmental reasons. That person there is an exact copy and clone of that person up there. So cloning as a process, not actually that complex. It's just reprogramming an egg with 100% DNA that you get from any cell, from any organism. Now, what's weird, just to throw out some weird stuff in this, I've cloned the man from up there and the woman's given an egg, and then there's been a surrogate mother and there's multiple people involved in this. What you could do, what could be really weird, is the woman at the top could be the subject. So we could take one of her cells with 100% of her DNA, also use her as the egg donor, so she gives one of her own eggs. We put a nucleus from her own cell into her own egg, reprogramming it. Also use her as the surrogate, so she gets implanted with her own embryo clone of herself, and she effectively gives birth to herself. Now that's really weird, and ethically, you know, there's explosions going off all over the place ethically as to how acceptable that is. But it's a possibility, and if you understand that that possibility exists, then you understand this process. Now, what if, though, you didn't just want to clone the individual, but you wanted to introduce something new, and you wanted to manipulate the original genetics to add something new, add a new characteristic? So what we're going to do, we're going to get rid of all this writing here to clear myself some space, and we're going to change the title over there to genetic modification. So this is taking the cloning technology we've just gone through, and we're going to jump it forward to an additional layer of genetic engineering. Now, what I've got here, if I've got the original guy next to me, I might want to make an improvement to him. So I take a second organism with a desirable characteristic. When I say a desirable characteristic, I mean it has a gene for a desirable protein. Now, in this case, I've got a plant. One amazing thing that plants can do is photosynthesis. We looked at photosynthesis in some previous videos, and essentially the point of photosynthesis is to take energy from sunlight and trap it in a molecule of glucose. Glucose being the food for almost everything on the planet. Plants or producers form the basis of almost every food chain. So the ability to photosynthesize and make your own energy source, make your own food, would be a massive advantage. So if I take the plant, I remove one of its cells, just in the same way we did with the somatic cell cloning for the subject. I take one of its cells, I identify and extract the desired gene that I want. Now, in this case, it's the gene responsible for photosynthesis. Now, photosynthesis actually requires lots of genes, but let's say it's just this one gene, and that's the gene for the production of the chlorophyll protein. Okay, if I've got that gene, what I can do is insert it into the subject's DNA. If I put it in that nucleus that we took from the original somatic cell, if I put the DNA into that nucleus, then it gets transferred into the egg, creating the zygote, the artificial zygote. So that artificial zygote now contains the chlorophyll gene. Now, if you allow mitosis to happen, you'll get an embryo that contains all the cells, and the clone will be green because it will produce chlorophyll. Now, it's not that simple, and of course the chlorophyll has to be produced in the skin if it's going to be hit by the sunlight to produce glucose. But what it means is that hopefully he's going to grow up to be an adult with green skin who could just go outside when they're a bit hungry or a bit low on energy and literally get solar powered, absorb that sunshine, create the glucose for themselves. Now, this is the basic essence of genetic modification, where you're modifying, not massively changing, just slightly modifying an individual to incorporate new genes. In this case, the chlorophyll gene to make a photosynthetic organism. Now, we've done it with humans here. I said before, it's internationally illegal to mess about with humans like this, but you could do this with anything. You could take the subject organism that you want, but you think, wouldn't it be better if I just have this little extra bit, and you can add that in. That's what we look at in the next genetics video. So from here, head to that video, which is genetic engineering 3 examples of genetic modification. So now that you've got the theory from this video, I want you to head to that one to look at some of the stuff that's already been done, increasing the nutritional qualities of crops, for example, and then look at some of the possibilities that genetic modification opens up. Now, it is, of course, a huge ethical and moral minefield of manipulating these genetics. We touch upon that in the next video, but only to a very small extent. But I want you to know how big and contentious an issue this is. But head to that one now. As always, please do subscribe to my videos if you haven't done so already. Like this video if it was useful to you, and go have a look at all the information that's on my Facebook page. Thank you.